principles and processes. And the chapter after this is the applications of the same principles and processes, right? So I'll just go quickly over what we did in the last class, basically what we talked about. Did you attend the first class of this last to
No,
last
sir.
class? No. So you technically missed the whole chapter, like till now.
Yes, sir.
Okay. So just to give you a quick summary before others join. So you know that biotechnology, it's derived from two words, bio and technology. Bio is associated with life, and technology simply means study of various techniques or developing various techniques through that study. So it is the process by which we use living organisms or the components of a living system to produce useful products for human beings. So basically the whole idea is that we use biological processes or products and use it for the welfare of human, right? So almost everything around you, mostly with respect to medicine and food, are a result of biotechnology, starting with vaccines, and they're a result of biotechnology, drugs. So biotechnology is an umbrella term. It cannot be pinpointed to one thing or the other. Wherever you develop a new technique or you use genetic tools to perturb the genetics to make something useful using a cell or a protein from the cell or any living component, that's basically biotechnology. So if in reproduction chapter we studied that IVF is one way of assisted reproductive technique, that's also a product of biotechnology. Okay. Gene therapy, this also I taught you in genetics chapter. When anyone has a genetic disorder that can only be treated by giving some or the other kind of gene therapy, there also we basically perturb or tweak the genes or the genetic makeup. So that also comes under the domain of biotechnology. Genetically modified organisms, whether they are plants or animals or microorganisms, and through which, for example, we have made high yielding varieties of crops, right? All these are under the domain of biotechnology. Is that clear?
Sir, yes. So I have a question in reproductive health. This one question.
You have a question in
Reproductive health.
Yes, please ask.
So in the surgical method called as sterilization, mainly tubectomy, we cut the tube, we cut the fallopian tube of a female and then we tie the knot, right? And that doesn't prevent the ovulation, right?
Right.
So won't the ovums get accumulated there?
Yes, of course. So the way you think, ovum is a cell, right? Any cell, and ovum dies anyways if you do not cut the fallopian tube. The ovum, when it is released, it's not living there forever. It dies within a few days, 2-3 days, right? So it will come, it won't get any passage to pass through to the uterus or to the ampulla isthmus junction where it can get fertilized, but it will stay there, and within 2-3 days it will die and degenerate. The next ovum will come and will follow the same fate. So it won't accumulate like in numbers. You won't have 20-30 ovums at the end of female's life or something. They will all die and degenerate. They are single cells, right?
Yes.
Yeah. Good question. Okay, so is this part clear to both of you? Good evening, Parker. How are you doing?
Good evening, sir.
Good evening. Aisha has also joined in. Good evening, Aisha. So in the last class, I basically focused on the core techniques on which this whole concept of biotechnology is based on. So basically, in the principles that biotechnology follow is either you make amendments or you change or you perturb the genetic material itself, in which case it is known as genetic engineering. In genetic engineering, we alter the chemistry of DNA or RNA itself by, by uh, creating mutations or by removing a base or, inserting a base or something like that, or by removing a whole gene out or putting a whole gene in through various methods. So basically, whenever you tweak or you change or you perturb the genetic material itself, that's called genetic engineering, right? You are engineering the genetic material. But apart from that, a whole lot of biotechnology depends on the process and developing the process itself. For example, for IVF, you have to take a ovum and a sperm from the male and the female donor and then make it fertilize in sterile condition in laboratory. And once that fertilization is done and it starts developing into a zygote, we implant it back into the female body. Now, to do all of that, we need to maintain sterile ambience. We need to develop um, tools and sophisticated machineries where we can do all of it without contamination. Even a bacteria should not come and contaminate, or any toxin or any protein or anything should not come and contaminate that whole experiment. So the thing is, maintenance of this sterile ambience is equally or even more important than engineering the genetic itself. So that's called bioprocess engineering. So these two are the pillars of biotechnology. Okay, either you engineer the genetic or you engineer the bioprocess around it, which is required to do the genetic engineering. And then I talked about plasmid. I introduced plasmid, and you all know what is plasmid, right? Plasmids are free floating extra chromosomal circular DNA in bacterial cells. So many strains of bacteria have plasmids. And these plasmids, the best thing is, yes, Parker, do you have a question?
No, sir.
Yeah. These plasmids, the most interesting thing is that they can self-replicate independent of the chromosome of the bacteria. So bacteria has a single chromosome, a long DNA thread, and it replicates. And we know, I've taught you in DNA replication, how does DNA replicates? What is the most important thing for a DNA to replicate? Yes, Parker.
The division of the strands.
Yes, so you mean opening of the strands, right?
Yes, sir.
The strand opens and then polymerase sits and DNA replicates. That's correct, perfect. But my question was more specific as to, can DNA replicate from anywhere, or like it replicates from one end and goes all the way to other end?
3' to 5'.
3' to 5', going through the DNA, like full length of the DNA? Like if the DNA is very long, right?
Yes, sir.
So does it go 3' to 5' throughout the length of the DNA, like starts from one end and ends at the other end?
No, it does not have the same ends.
No, then what happens?
DNA is just a long chain.
Right, it's a long chain, it's double stranded, and to replicate it has to open up, and in between, following the rule of semi-conservative method of DNA replication, DNA makes its copies. But what I was asking is, does it replicate anywhere in the cell or from any part or any sequence or any gene? The answer is no. DNA has specialized regions which are called origin of replication sequences, and DNA replicates from that point only. So there are multiple of these ORIs, origin of replications, in in the genome. So for any DNA to replicate, it needs to have its own ORI, origin of replication. ORI comes from the word origin. Plasmids have their own origins of replication. That's why they can independently replicate without depending on the chromosomal DNA. Now, this is very interesting because through plasmids, bacteria get all different kinds of abilities that their genome does not provide. For example, antibiotic resistance. So this antibiotic resistant gene is present on a plasmid, and this plasmid can replicate and then transcribe and translate and form the protein that gives the bacteria resistance against antibiotics. Okay. And this experiment was done, and it was found that plasmids actually do contain these antibiotic resistant genes. Then I came to discussing two scientists, Herbert Boyer and Stanley Cohen. Yes.
So sorry. So in this experiment, you are transferring the gene of the plasmid from Salmonella to E. coli, right?
Yes.
So won't Salmonella be devoid or lacking the gene?
Yes, it will be. So we are removing the plasmid from one strain and we are giving it to another strain. We are culturing
So
both the strains in the lab. Yeah.
will that impact Salmonella because it's getting devoid of
Yes,
the gene?
yeah, Salmonella will no longer be showing antibiotic resistance and will die when it will encounter that antibiotic.
Then what's the point of doing this?
Because we want to prove that the gene is not the gene is not on the chromosome. The gene is on the plasmid. And if you can, the point of doing this is, that's a very good question. What's the point of doing this? Are we just killing one bacteria and saving the other? That's not the idea. The idea is, can we develop a method through which we can harvest these plasmids, and specifically these plasmids from bacterial strains that have it, then change it or modify it or engineer it according to our will, and then transfer it into other bacteria that is not pathogenic, but will replicate and also replicate and transcribe and translate our gene of interest for us. So this whole thing was done to develop this biotechnological process where we could harvest plasmids from one bacteria without harvesting the genome, because we don't need that. We just need small circular plasmids. Then we could insert it into another bacteria, and the ability that the previous bacteria had, now the new bacteria that did not have that ability gets this ability. This proves that this is a very good technique we have figured out. Now all we need to do is go one step ahead. We will harvest plasmid, or we will create. Nowadays we create our own plasmids. We don't rely on bacteria and harvest plasmids from them. We can synthesize plasmids, artificial plasmids. And I have talked to you about some of those BAC and YAC, right? Yeast artificial chromosome and bacterial artificial chromosome. BAC and YAC, they are used as vectors. Did I talk about that? Do you remember when we? Yeah. So those are the ways by which we can create our own plasmids. We can also put any gene that we want to be translated and form protein. And I was giving the example of insulin in the last class. So this whole process is done so that we can insert insulin gene in a plasmid, put it in bacteria, let the bacteria produce insulin for us, insulin protein. We will purify the insulin protein and save millions of diabetic patients' life. Do you understand that?
Yes.
Does that answer your question, Hiram?
So, as you've drawn that figure, you've drawn 3 plasmids in the Salmonella. So if you take out one gene, we have the rest 2 plasmids. So that won't be like detrimental to the Salmonella.
Yes, so plasmids, right? If you are devoiding a bacteria of all the plasmids, then of course it will affect. But yeah, plasmids also, like I said, they are self-replicable. So you can remove one and leave the other, but normally it doesn't happen. When you are harvesting from bacteria, bacteria gets killed in the process. You have to burst open the cell to get the plasmids out. Yeah. But technically what you're saying is that it has multiple plasmids. Even if it transfers one plasmid to someone else, it can have other copies. But when we are harvesting, we are not worrying about the lives of bacteria, right?
Yes, sir.
Yeah. Yeah. Then I talked about cloning. What is cloning? It is making multiple identical copies of any DNA template. When we make copies of our plasmid, we are actually cloning the plasmid, and that plasmid contains our gene of interest. So that way we are cloning our gene. So that's called gene cloning. So gene cloning is nothing but putting a gene of interest in some organism where it make multiple copies of it. Okay. We can also do cloning using PCR technique, which I will teach you about in this chapter. That is a more targeted way of making doing cloning of any fragment of DNA. But the other one is putting it in a plasmid and sending it in a bacteria. Okay. Another important thing that I discussed in the last class, I'm just giving you all this because those who have just joined now and missed the last two classes won't feel lost in today's class then. So, and also please don't miss classes, because then I'm in a dilemma in the fork road, whether to discuss everything that I have done before or to go ahead to complete the syllabus. So it's not like next class you join and I can just start from there, because that doesn't help you, right? That's not the point. So restriction enzymes is another thing. Restriction enzymes are molecular scissors. And these enzymes are present, again, they were isolated from bacterial strains. They're of two types, exonucleases and endonucleases. The word exo and endo speaks about it. Nuclease means that degrades nucleic acid, right? Now, exonucleases work from outside, like from the sides of a DNA chain. So they kind of start chewing up the DNA, like you can see in this image. They chew up the DNA from one side, from outside to in. Okay. Whereas the endonuclease cuts the DNA at any specific sequence um, in that sequence, right? So that's called endonuclease. Now, having known that there are exonucleases and endonucleases, exonucleases do not work on plasmid. Can anyone tell me why? Why will an exonuclease not work on a plasmid?
Because that circular
Plasmid is
Yes, plasmid is?
it is a ring.
Yes, it is a circular fragment. Also, Iram said the same thing. Yes. So plasmids, being circular in nature, do not have loose ends or ends at all. So exonucleases cannot work. So whenever we work with plasmids or vectors in biotechnology, we mostly focus on endonuclease, and that is the tool that we use for cutting anywhere and inserting any piece of gene of interest or something. So endonucleases are all I'm going to talk about in this chapter from this point onwards. And whenever I say restriction enzyme, just by default understand that it's endonuclease, right? Now there are almost around 1,000 different kinds of endonucleases that we have uh, discovered from different strains of bacteria. And endonucleases are a great tool because they provide specificity. Each endonuclease will cut the DNA at a specific location, very, very specific sequence that only that endonuclease will recognize, and no other endonuclease will recognize that. So no two nucleases, endonucleases, will cut at the same site. So that is a very good uh, package for us. Like we can choose which endonuclease we choose from and cut our plasmid accordingly, right? And then I talked about how these endonucleases are named. So we took two examples, HIND2 and EcoR1. So HIND2 or EcoR1 in the name of these nucleases, the first alphabet is basically the genus of the bacteria from which these nucleases come. So HIND, so H is for Haemophilus, IN is for influenza. So next two letters, which are in small, talks about the gene species, and then last letter is the strain. So it's Haemophilus influenza strain D, and the Roman numerical often tells about the order of isolation of this endonuclease from that strain. So HIND2 means it's isolated from Haemophilus influenza D strain, and it was the second restriction enzyme to be discovered or isolated, not discovered, but isolated from the strain. Okay, same is for EcoR1, E for Escherichia, CO for coli, R is the R strain of E. coli, and 1 is it's the first to be isolated from the strain. Okay, and just a fact that HIND2 was the first restriction endonuclease to be isolated and used in biotechnological process. So just remember that this is the first restriction endonuclease that we got, that scientists figured out. Now there is a catch. There's a 2 in the name, which says that it was the second one to be isolated. And I'm saying that this is the first restriction endonuclease. So I think it makes it very clear that the first one would have been an exonuclease, right? Not an endonuclease. Make sense?
Yes.
Yeah. Okay, perfect. So having said that, in today's class, what I want is to take it one step further and talk about what are the characteristic features, or what is important when we use these tools, restriction endonuclease and the plasmid, for biotechnological processes. Okay. Okay. So if you remember, sorry, is it paused? Is the sharing paused?
No, sir.
You can see the screen, right?
Yes, sir.
Okay, perfect. So if I ask you a question, we have restriction enzymes now. Okay. We also have plasmids, right? Now what we need to do is we need to cut, correct?
Sir?
Yes.
Sir, are you writing something?
No, I'm not writing at the moment. I'm asking a question. I'm going to write in some time. So the question is, we have the scissor. We also have the enzyme. Sorry, the enzyme is the scissor, and we also have the plasmid, the vector. Now we have to cut, right? But my question is, if just cutting a plasmid will make it linear, right? If I make one cut in the plasmid, it will make it linear, correct?
Yes, sir.
So making one cut in the plasmid leaves how many fragments?
2.
1. That's the point. It's not linear to begin with. So do not confuse a plasmid with a long thread of DNA. If you make one cut in a plasmid, right? So this is a very simple thing that often, we don't pay attention to, and we lose marks in entrances. If you make one cut in a plasmid, it gives you one fragment, which is a linear now. So one cut makes a circular plasmid linear. Still just one fragment. Okay.
Sir, can you explain that again?
Yep. So let's say this is a plasmid. Okay, it's double stranded. For the sake of simplicity, I'm not making the helix. This is also helical. DNA is always helical, but just let's assume that this is a double-stranded plasmid, correct? Now, if I make one cut here, it will just open the plasmid, right? I will get a long, from a linear, it will, so from a circular, It will become a linear fragment, but it will still be just one, right? Just like this.
Yes,
Do
sir.
you understand? Yeah. So this is what I'm saying. But if I was making one cut in a linear fragment, if I make one cut here, I will get two fragments, correct?
Yes, sir.
Yeah. So in a plasmid, we make one cut and we just get one fragment. And this is where it becomes important for us to know how does it work. Now, if you all can come to this
So they don't
figure.
attach back.
Yes, that's what I'm going to talk about. So come to, where did it go? Let's see. Yeah, can you see the image that I have attached?
Yes, sir.
Yeah, this is from your book only. So this is how a plasmid works. Sorry, this is how a restriction enzyme works. So this is for restriction endonuclease. So from now onwards, as I told you, when I use this word restriction enzyme, just remember that I'm talking about endonuclease, not exonuclease. Okay. Now, just pay attention. So the enzyme basically cuts both DNA strands at the same site and at the same time also. So this is your vector DNA and this is your foreign DNA that you want to insert. Okay. So this will go on being like this. Basically, imagine this is a part of a circular plasmid and this black thing is what you want to insert. Let's say this is the gene of your interest, insulin or something that you want to put in a plasmid. Now, the way to do it is you will cut open the plasmid, right? I made a cut, now it's open. Make sense?
Yes, sir.
And I want to put my gene of interest here. But how I'm going to fix these ends was always a problem. So this was one issue that got solved when we discovered restriction endonuclease. Now, what do restriction endonuclease do very beautifully is that wherever they cut, they always produce sticky ends. Now, what do I mean by sticky ends? Is that, let's say we are using EcoR1. This is an example of EcoR1. So EcoR1 cuts the DNA between bases G and A only, when the sequence is GAATTC. So this is the sequence that EcoR1 recognizes. No other endonuclease will recognize the same sequence. Only EcoR1 recognizes the same sequence. And whenever it recognizes this sequence, so wherever in DNA this sequence is present, with the help of EcoR1, you can see here it is GAATTC. With the help of EcoR1, you can cut that DNA between G and A, which means exactly at the beginning. So this is the middle. It will either all plasmids work like this. Sorry, all restriction endonuclease work like this. Either they cut on left or right side from the, they never cut at the middle. If they cut at the middle, you won't be able to form a sticky end. You understand? Now, how does a sticky end gets formed? Let's see here. GAATTC. So on the opposite strand, it will be, what will be on the opposite strand?
CTTAG.
CTTAG. Perfect. Now, so this is the sequence. So EcoR1 will recognize it in this direction, and On the other strand, eco R1 will recognize it in this direction. Am I correct?
Yes.
And where will it make a cut? Let's say this is the 5' to 3' strand, and this is the 3' to 5' strand. And we know that it makes the cut between G and A. Okay. So where will it make a cut on 5' to 3' strand? Here, right?
Yes.
And on the 3' to 5', it will make a cut here, correct? Between G and A.
Yes.
And then it will remove these two strands. So when you will remove it, you will be left with something like this. This is, okay. So on one side, you will have G paired with C, T, T, A, unpaired, right?
Yes, sir.
And on the other side, you will have A, T, C unpaired, and C will only be paired by G, correct? So this
Yes, sir.
region which is unpaired here and here, these are empty, vacant paired, like vacant pairing is there. So the moment I again bring them closer to each other, it's like a magnet, the ends of magnet, you understand?
Yes, sir.
Yeah. So the moment you will bring these two closer, what will happen? They will again click.
Attach.
Yeah, they will again attach, click back to each other. Make sense?
Yes, sir.
Yeah. Now, if I want to not, if I cut the vector and the vector attaches back, it's like I opened a ring and it got closed again. It's a failed effort, right? I don't want that to happen. What I want is, I want my gene of interest to attach between here, one end here and other end here. Basically, what I was representing here, right? So I want to use these sticky ends to insert my gene of interest between the two ends of plasmid. So it again becomes circular and closed, stable, and I can insert it easily into a bacteria. So it will, it's like I added something to the plasmid, but this time what I added is what I want to be translated, replicated, transcribed, and translated. Does it make sense to all of you?
Yes, sir.
Now, how do I do this? One way to do it is you use, and the only way to do it is you have to use the same restriction endonuclease to cut the plasmid and also to cut your gene of interest from the whole genome of the organism. Suppose I am cutting a gene of my interest. Again, let's go back to insulin. So I want to cut the insulin gene from human genome and put it in a plasmid. So I will use, first I will have to see, can I cut out insulin gene using EcoR1? If insulin gene has this sequence GAATTC in the middle of the gene, can I use EcoR1?
Yes.
No, I cannot, because it will cut my gene into two parts. I want the gene intact, right? If I cut my gene into two parts, then what I will be inserting is a half gene. It will not be a fully complete gene. And when it will translate, it will produce a half protein that will not fold properly or will not function properly. So it is of no use to me. Do you understand? So the sequence that restriction endonucleases recognize should not lie in the middle or somewhere in the gene of my interest. It should be a little left or a little right, like before the uh, promoter and after the terminator site of the gene. Make sense? Does it make sense to you people?
Could you please explain that part again?
Yeah. So this is my insulin gene. This is where the gene starts, and this is where the gene ends. I want to put this gene into a plasmid. Make sense?
Yes, sir.
Yes.
Suppose I'm using EcoR1. So EcoR1 recognizes one sequence, which is GAATTC. If GAATTC is present in my gene, and if I use EcoR1 will cut my gene only, right? And my
Yes,
gene is destroyed. What I want is, I want to cut my gene. So if this is the start site, I'm okay cutting it a little here, and I'm okay cutting it a little here, like as close as I can. But of course, I will not be able to cut exactly the sequence of the gene, right? I don't want to disturb the gene. It's simply like you are cutting out a sticker. You drew something on a paper and you cut it out. You try to avoid the boundaries, right?
yes,
You
sir.
made a hut. Now you want to cut it out to make it a sticker. So will you make your scissor go like this or like this?
No, sir.
No, right? You will very, very specifically try to leave a little bit of space and cut it like this, right? Because all you want to protect is what is inside. Same is there for gene. That's why we have hundreds and thousands of restriction endonucleases. First, I will check which nuclease will be the best nuclease I will use. So it will not cut my gene, but it will not cut my gene from within, but it will cut little left and right flanking. It will leave some flanking region, and I can take out the gene without destroying the gene or without damaging the gene. And then I can put it into the plasmid. Does it make sense to all of you?
Yes, sir.
Yep. Aisha, Yeah. Iram, are you clear? Since you asked the question.
So, but in this experiment also, the same thing is happening, right?
What is happening?
The E. coli is cutting it from the middle, right? From the A region and from the G region. No, sorry, from the G region.
Yeah. Sorry, I didn't understand your question. E. coli is cutting from the middle means?
So as you said
Eco
about the
R1.
eco R1, sorry. Sorry, eco
Okay.
R1 is getting, yeah.
Right. I got it. So what is a gene? A gene is a sequence, right? It starts with AUG, there'll be TTAACCG, something like that, right? A full gene. If you use Eco R1, it will recognize this sequence. And if that sequence lies within your gene, the start and the end of your gene, then it will cut your gene only. You don't want that. You want a restriction endonuclease which recognize some sequence which is at the end of your gene, somewhere here or somewhere here, right? So your gene remains untouched. And then you cut from here, take out that whole piece with your gene inside. Of course, you will have some extra sequences, but we don't care about that. Till the time our gene is intact, it will get translated properly. It will get replicated, transcribed, and translated in a plasmid inside the bacteria. And we will get a full protein. Make sense?
Yes, so that's what in the vector DNA we have the GATTC part at the end. So it is unaffected, right?
In the vector DNA, we have what?
The GAATTC part, as it's in the figure.
There is no
It's at the
end
end.
or beginning. How can you say something is at the end in a circle? What is an end in a circle? Vector is plasmid, it's circular, right?
Mhm.
So what is at the end of a circle? There's nothing at the end, right? We just need to have GAATTC in the vector somewhere, and we need to have GAATTC outside the region of our gene of interest somewhere. Then we can use EcoR1, because we will cut our gene out, we will cut open the vector, we will put it inside because it has sticky ends. Yeah.
Yes, sir. Yeah.
Yeah. What I also, yes, Parker.
No, sir, go on.
Okay. So this is how, just look at it. It produces sticky ends, and then this part goes and clicks on this part, and this is how two DNA join together. This is your vector, and this is your gene of interest. This will be round like this, and this will again, so it will make a whole circle. Okay. Perfect. And then it's ready to be, this is called a recombinant DNA, because we have engineered this DNA by cutting some pieces from somewhere, cutting some pieces from somewhere, and putting it all together. This is not a natural DNA. So it's called a recombinant DNA, that artificially recombinant DNA. And this is the whole crux of biotechnology. Anyone has any questions still here? Any doubts?
Yes, sir.
Yes, Parker.
Yes, sir. The restriction endonuclease, sir, this sequence GAATTC, it wouldn't repeat like in a plasmid. Like for example, there wouldn't like this.
Yeah, that's a very good question. So what you are trying to say is, in a plasmid, let me find it. Another figure. Yeah. Come to figure 11.2. Those who have older books, you can come to the figure 11.2 in your textbooks. If you have the newer textbooks, I think it will be 9.2, I guess. And did you find the figure?
Yes, sir.
Yeah. Is this the figure that you found?
Yes, sir.
Yeah. This is a diagrammatic representation of the whole process that we were talking about. And I will address your question using the same figure, okay? Now, look here. So what we were doing is, this is our foreign DNA. This is our gene of interest, right? This.
Yes.
Let's say this is the gene that I want to replicate and get multiple copies of, or the product of. Now, it is very obvious that gene must be starting somewhere here and must be ending somewhere here, right? And this is the
Yes, sir.
extra region that I got to be safe, so that the gene is safe in the middle. And whatever endonuclease I used recognized a sequence here and recognized the same sequence here. So it had the same sequence before the gene started, and it had the same sequence after the gene ended. If I would have used another endonuclease, it would have cut somewhere here, and that's not what I want. I want this. And then you use the same restriction endonuclease to cut your vector DNA plasmid also. Now, what Parker is asking is, the vector will also have the same sequence that was present here and here in it. Then only the endonuclease can cut it. And that's why nowadays we design our vector plasmids, and we can put as many sequences that are recognized by different restriction endonuclease as we can. So it is like a fully formed package for you. You buy it from biotech companies, and then you decide which endonuclease you want to cut it with. It will have 5 or 6 multiple sites where different endonuclease can cut. Now, Parker's question is, let's say this is the site where it cuts, okay?
Yes, sir.
This is the site recognized by restriction endonuclease, RE. Now, for each and every restriction endonuclease, every plasmid should just have one site where it can cut. it should not have more than one site. Why? Why do I say so? Aisha, Iram?
Because then it will not be a linear.
It will be a linear.
It will
The
have
moment you,
more than two fragments, right? I mean,
it
two
will have more
fragments.
than one fragment.
More
Yeah.
than-
The moment, yeah. If it has just one cut, like here, let's see, it cut here, and then it is one fragment. And you can insert something in between here, right? From between here to here. But if you have two such sites that each restriction endonuclease can recognize, let's say the another site is here, then this plasmid is a waste, because the moment I will treat it with restriction endonuclease enzyme, it will cut here and here. And how many fragments I will get? Two fragments. And if I have two fragments, I cannot put it back into a ring. I cannot make it back a plasmid. Make sense?
Yes, sir.
So I only need to open it at one place so that I can insert. So you can see here, this is what they have inserted here, right? They have inserted something, they close the plasmid, and they have put back this plasmid into E. coli. And this process is called transformation. Joining of DNA to plasmid is called ligation. So first process is restriction, or sometimes in book they call it restriction digestion. You are cutting your DNA, you are cutting the plasmid. So it's restriction digestion. Then you want to ligate the DNA of interest with the plasmid. This is called ligation, right? Which is joining the gene of interest or the foreign DNA. When I say foreign, the word foreign is from the perspective of plasmid. Make sense?
Yes, sir.
So for plasmid, it is something which is not its part. It's a foreign material, alien DNA. And then the third is transformation. Why I call it transformation? Because I am transforming this bacterial cell here by putting a plasmid inside it, which contains some genes that is not the bacterial gene, right? Bacteria did not have these genes naturally in its genome. I am sending it through a plasmid. And then because plasmid can self-replicate and then transcribe and translate independent of its own, of the genome of bacteria, it will behave like an independent mini genome and will produce more and more proteins that I have sent in the bacteria. The bacteria does not naturally or natively make those proteins. So this process is called transformation because we have transformed the bacterial cell by inserting inside it a recombinant DNA. Make sense? Yes.
So ligation is the step where you stick the foreign DNA to the plasmid, right?
Yes.
But what's the necessity of it? Because it anyways produces sticky ends, right?
Right. It produces sticky ends, but to be 100% sure that, so, sticky ends are also between the cuts of the plasmid itself. What is avoiding the plasmid from self-clicking back? Nothing, right? So what normally happens is we take a tube, and in this tube you have your cut plasmids, which will look like this, C, right?
Yes.
I'm making it C so that you understand. Basically, they will be linear. Now, what's preventing them to just click back and become circular again, right? They can do that. Yes or no?
Yeah.
Do you understand? Yeah.
Yes, yes.
So what happens is, instead, let me make it back again. See, all of them are back. Yes. And we put, along with these plasmids, cut plasmids, we also put our fragments that we want to ligate with the plasmids. Okay, make sense? These are our fragments. And to make this process more efficient, we use an enzyme called ligase. Now, ligase will favor joining of one plasmid with our gene of interest, and when it will close like this. But of course, some of them will just click upon themselves and close back. Do you understand?
Yes.
So this process is to ensure that we get enough plasmids with our gene of interest ligated in it. That's why we need to do this step in a more controlled manner. We cannot just open it, mix it with our gene of interest and shake the tube and all is, and just pray that all is. That way also, some will of course get our gene of interest into the plasmid, but to increase the chances, probability, and the number of these plasmids that are transformed, we do ligation properly using an enzyme ligase. So ligase, whatever, when we use the enzyme ligase, it favors ligation of foreign DNA with the plasmid over the plasmid just ligating to itself. Does that answer your question, Hiro? So that is what the need is.
Yeah.
Yeah. Because we are doing all this expensive business to get our gene inside a bacteria. Okay. Even after doing it with DNA ligase, let me tell you, it's never that 100% of your plasmids will get your gene of interest ligated in it. Some will just close back. Okay. But the good thing is, even if we have 50%, 60% of them, when we will insert all these plasmids in the bacteria, plasmids replicate. So 1 will form 2, 2 will form 4, 4 will form 8. Make sense? So the numbers will increase eventually.
Yes.
Yeah. Okay. Now, after transformation, the next step is culturing, cell culture. We culture these bacteria into big bioreactors, big vessels. Why? Because we want them to grow into millions and millions and billions and billions. And each bacteria will then produce the protein that we have inserted the gene for. And after that, when there are billions and billions of bacteria, all full, then we will take all these bacteria, burst them open, kill them, because we don't need them anymore. They are like brooding pouches for us, for our protein. All the protein we will isolate using a technique called protein pull down assay, where we can kind of make a bait, a molecular chemical bait. Those things are not a part of your syllabus, but to understand, you can actually, from a pool of proteins, because when a cell opens, there are lakhs and lakhs of different kind of proteins in a cell, right? So you can pull one type of protein from all other proteins in your cell by making something that only binds to that protein. So there are protein binding proteins or protein binding RNAs that we can use as bait. It's like fishing in a pond. If you want to fish out very small fish, your bait has to be small and something specific that fish eats, right? You cannot fish all the different kind of fish with the same bait. So it's the same concept. Then that's called purification of protein. So after this culturing part, the fifth part and the sixth part is purification. And once we have purified our protein of interest, then we go for large scale production, large scale packaging, or what do you call it? Large scale, production only. So to keep that protein stable, we have to dissolve it into some kind of concoction or chemical which will keep it stable, like insulin that we got. Let's say this was all done for insulin. We need that insulin after purification to be functional, viable, and in its proper shape. So we will dissolve it with something where the pH and all maintains the insulin in its proper form. And then when we will inject it through injection into a person's blood, that insulin will be ready to do its function. It will go and bind to different cells in the body and will allow the cells to take up glucose from blood. Do you understand what's happening here? People, just look at this figure and let me know if you understand or if you have any doubt. Anyone has any doubts?
So,
yeah,
about having two sides of recognition in the plasmid, you will get two fragments out, right?
Right.
So why don't you get two genes or two copy of gene of interest and attach to it?
Because you're cutting the plasmid with the same endonuclease, right? What you are saying is also done. And often we never just put one gene. We always put two genes. One gene is our marker gene. You also need something which I'm going to tell you the next. You also need something to be a marker. When you are doing all this, you are not seeing anything actually. You just mix one thing with the other, one thing with the other, thinking that everything is happening fine. Molecular biology is very boring and tedious because you don't get to see anything with your eyes. You just believe that what you are doing is technically correct and you do not have any contamination in your samples. The end result will only tell you whether you have succeeded or not. So you will never get to know that your experiment is failed or failing most of the times midway. You don't know that. And that is also very, very patience testing for molecular biologists. They only get to know of the failure most of the times at the very end of the process. So what we do, we also put one marker gene that will tell us at the end of the process that visually we can know that, okay, whatever we did has worked. So we often put more than two genes in a plasmid. But for that, we need two different restriction endonuclease to cut the plasmid in a sequential manner, one by one. You insert one by cutting it at one place. So suppose for gene 1, I used HIND2. So I will use HIND2 to treat my plasmid and my gene of interest, then ligate it. Then again, after ligation is done, I will take EcoR1, again treat my plasmid and my genome with that, again ligate. So it has to be sequential, because once simultaneously, if you cut two fragments, those two fragments will be lost in the ocean of molecules. They will never come back together and get clicked. You understand?
Yes, sir.
Nothing is holding those two fragments together, no? Does it make sense to you? And the sticky ends are everywhere. So you have to make sure that your plasmid remain intact, of the same shape and the same size. So you have to do it by cutting it once, putting something in, then cut again, put something in, cut again, put. That way you can put 10 things in it, not a problem. That's called a cassette plasmid. Cassette means, you know, a cassette. A cassette has multiple songs in it at different locations of the reel, just like that. We will talk about it in detail, but does that answer your question? Is the process clear to you in your head? Hiram?
Yes.
Okay, perfect. Now my next question to you is, this is done, let's say. What do we want to achieve the next? Let's understand first what are the features of a vector. So what basically is the word, what does the word vector means? Come again, tell me. Vector, we discussed it yesterday, but I think I just discussed it with Rabia or someone. Was it Parker?
Yes, sir.
Yeah. So why don't you answer, Parker? What is a vector? What do we mean by a vector? Vector. You must have heard this word vector. A vector is anything like in biology. You must have heard this word in physics, vector quantity and scalar quantity. There also it's kind of, you can make similarities in the concept. A vector is something that carries, something from one place to another. And in physics, it's something that has a direction, right? Not just magnitude. So in biology, we use the term vectors for all those things that are not themselves the causative agents, but they carry something in it and transfer it from one system to another, which is the causative agent. For example, for the disease dengue, malaria, we say that mosquitoes are vectors. Yeah. Because if you see what causes malaria, if I ask you the question, what causes malaria?
Mosquitoes.
Mosquito don't cause malaria, because if mosquito causes malaria, every time you are bitten by a mosquito, you will get malaria. Do you get malaria every time? Yes. So the causative agent of malaria is by observation, people in earlier periods also thought that, oh, mosquito causes malaria. But then the question is, we are bitten by mosquitoes multiple times, but we don't get malaria all the time. But sometime we are bitten and we get malaria. So it's not mosquito, it is a protozoa called Plasmodium, the most common Plasmodium falciparum or Plasmodium vivax. So it's Plasmodium that causes malaria. Now, mosquitoes act as a vector and carry these Plasmodium in their saliva, in their salivary glands. And if a mosquito is already having this Plasmodium, when that mosquito bites you, before they suck the blood in, they kind of release some fluid, which is their saliva, a very tiny amount, in our body first. And that fluid contains anticoagulants, because one property of human blood is the moment it comes out from your vessels, it clots. Yes. So a mosquito is basically intelligent. So if we just put a syringe in a human blood vessel and allow the blood to drip out, the moment it will drip out, after some time it will clot. It will clog the needle itself. But it never happens when someone takes our blood sample. It never happens when someone is getting drips through the blood. Drips through IV. It never happens when mosquitoes are sucking our blood. Why? Because all these things function on the principle of anticoagulants. The injections that are used to take our blood samples on the inside are coated with a chemical which is anticoagulant. It does not allow the blood to clot when it is inside that needle or inside that vial. Okay. Similarly, the proboscis of mosquito that goes inside our skin and sucks the blood is also coated with anticoagulant chemical. And that is exactly what it releases first, so that the nearby region of the blood, nearby area, does not start clotting, and it can very, very comfortably sip in all the blood that it wants. And while doing that, it also transfers some plasmodium in our body, and that causes malaria. So that's called a vector. Here, the plasmid is a vector because it is carrying our gene of interest into some another organism where it will form its protein. Okay, cool. So the word plasmid, origin of replication, cloning, all this is clear to you. Restriction enzymes, the plasmid acting as a vector to transfer the DNA which is attached to it. All this is clear to you? Yes, no?
Yes, sir.
Okay. Okay, now write down a few things before we go forward. About restriction endonuclease, write down. Restriction endonucleases, like in this restriction endonucleases, like in this case here, restriction endonucleases function by recognizing functions by recognizing a specific sequence in the DNA double helix. In the DNA double helix. Okay, okay. Next point, it binds to that sequence. It binds to the sequence and cut the two strands. It binds to the sequence and cut the two strands at specific points in their sugar phosphate backbone. So this is where a restriction endonuclease cuts. It cuts between the sugar phosphate backbone. It doesn't cut between sugar and the base. No, okay. So this is one neat question. Restriction endonucleases cut between pentose, sugar, and phosphate molecule, phosphate and nitrogenous base, sugar and nitrogenous base, all or none, something like that. So it cuts between sugar phosphate backbones. Because once you cut the backbone, the ladder is cut. The helix is cut. Between the bases, it is hydrogen bonding. So that you don't have to cut. You can just open it once the backbone is cut. Another interesting thing about restriction endonuclease, just write this thing also down. Every restriction endonuclease recognizes a specific palindromic sequence in the DNA. A specific palindromic sequence in the DNA. Now, what is a palindromic sequence? A palindromic sequence. The word palindrome is an English word. You know about the word palindrome? What is the word palindrome means? Let's see here. Palindrome is a word which, when you read from left to right or right to left, it spells the same. Okay, like the word Malayalam. You read it in this direction, it spells the same. You read it in another direction, it spells the same. Right? Do you understand? So this word is a palindrome. Similarly, you will always see, look at the ECO R1's recognition site, GAATTC. So for a DNA, the direction is of the strands. So when you go in 5' to 3' direction, you read it as GAATTC. In 3' to 5' strand, it's again GAATTC from the other end. So that's why it's called a palindrome. Because DNA is not a single strand, DNA is a double strand. So when I say palindrome, it has to be one strand will be one side and the other strand will be another direction. Okay. This also gives us a fair idea when we look for sites in the DNA that can be cut using restriction endonuclease. We often look for palindromes. Okay. Okay, cool. So this part is clear. And you can write down for a DNA, what does a palindrome means? That it reads the same, the sequence reads the same on the two strands in 5' to 3' and 3' to 5'. The sequence reads the same on both the strands. Now let's start an experiment of our own. Let's say we want to do some science with these restriction endonuclease. Now you understood what a restriction endonuclease can do. But one question is, suppose first thing that I have to do is I have to isolate the whole genome, the DNA from any organism. Suppose I want to take out insulin. I am taking this example of insulin throughout because that is exactly what is in your syllabus. So let's keep it in habit. So for that, I need human cells. I can take human cells from anywhere, like a tissue sample from pancreas or somewhere. And then just like you did your DNA extraction from banana, strawberry, and everything in your schools, using a more sophisticated version of that technique, we will isolate DNA without any contaminations. And now we know how can we isolate DNA, right? We studied it in chapter 5. DNA isolation. Everyone is clear with DNA isolation process?
Yes.
You open the cell first, then you open the nucleus and you take out all the nucleic acid, but you only want DNA, not the RNA. So what will you do? Because RNA is also nucleic acid. If you just want purified DNA and not RNA contamination, what will you do? You will add enzymes that degrade RNA and not DNA, called RNases. Right? Remember every MacLeod and McCarthy's experiment? Don't keep forgetting the previous things. You have to make a bridge between concepts. So we can add RNases that will specifically degrade RNA and keep the DNA intact. Once you get all the DNA, you need to get one gene from that whole genome. That is a daunting task. It's like one brick in the whole building I'm interested in. So to get that brick, we know that now we have molecular scissors, restriction endonuclease. I will look at the sequence and I will decide that, okay, if I use this particular restriction endonuclease, it will cut a little bit left and a little bit right of my gene, which will work for me. And I will get my fragment. But how will I separate that fragment from the total mess that I have created? I have a full genome where I have made some fragments and I need to get just my fragment out so that I can ligate it with the plasmid in order to send it in a bacteria. How will I do that? Do you understand the question? How to separate the DNA fragment of interest after cutting it with restriction endonucleases. Any idea? It's a basic problem solving question. What are the ways? Simple. So I have a bucket full of balls of different size. Okay. Now I am asking you, from this whole bucket, I only need all the balls that are 1 inch in diameter. I don't need the smaller ones. I don't need the bigger ones. I need all the balls which are 1 inch in diameter. Similarly, for our gene of interest, of course, we know the sequence length, right? We know how long is a insulin gene. We know how long is any other gene. So we know the length of the fragment, but the problem is that fragment is present with all other 1,000 different kind of fragments in a mess. How do we do that? How will you sort out all the balls of 1 inch diameter from a bucket full of different balls? You cannot handpick. You can handpick the balls, but you cannot handpick DNA. So let's say handpicking is not a good option. Any other thing you will do?
Sediment.
Sorry? It's a good. Go ahead.
I'm
Go
just
ahead.
guessing. Sedimentation.
Sedimentation. That's a very good guess to begin with. But when you do sedimentation, the bigger balls will sediment first. Right?
Yes, and the smaller one will be at the top, so you can get the smaller fragments.
But we don't know which is the smallest one. No, we just know we want 1 inch balls. There are thousands of different sizes of ball. The smaller one could be just 1 centimeter one. That is not what we want.
Or you can just stain that.
Stain that. Yeah, that's a good idea. But how will I stain a bucket of balls? Suppose I put a color from top, it will stain all the balls. How will I stain just a 1 inch ball and not the others? Right? For staining it separate individually, I have to hand pick it. First thing is, again, hand picking we can't do for DNA. But something like you said, we have to isolate them on the basis of size only, size of the fragment, right? For that, a very interesting process has been discovered and developed called gel electrophoresis. This is the answer. And I'm sure your schools must have taught this, because this is how we can do all our biotechnology. Gel electrophoresis. It uses the concept of electricity and charge. Now, what happens in this is, we know that DNA is a negatively charged molecule, correct? People say yes, keep saying yes or no if you want to understand this.
Yes.
I'm sure in your schools, either you didn't pay attention or they did a very bad job teaching this concept, so you don't know about it. But your aim should be, once you leave this class, you know what you were not knowing before. So DNA is a negatively charged molecule. And if there is a small fragment of DNA versus a large fragment of DNA, the negative charge will be higher here than here, right? Because the negative charge comes from phosphate group. So this will have, let's say, 10 phosphate groups. This will have 50 phosphate groups. So more negative, correct? Make sense or no?
Yes.
Yeah. So now, if I put these two molecules, one is the smaller one and one is the bigger one, and apply a electrogradient force, like if on this side I have a positive terminal, sorry, it is negatively charged, so it will be attracted towards positive. So this side it has negative, and this side it has a positive terminal, and I've connected it with a battery. And of course, it is in a solution, so otherwise DNA will be fried up. So in a solution, if I make this gradient, there will be an electromotive force on each molecule of DNA, correct? Simple physics. And to which side the DNA will move?
Positive.
Towards the positive side. Now, whether it is a smaller one or whether it is the bigger one, they will both move towards the positive side. Now, if I add one more layer to it, I am done with, I am good to use that as a method to isolate DNA fragments. So there is a race. In that race, I am allowing the DNA to move through a matrix. So it will be a gel. That's why it's called gel electrophoresis. So I make a gel. Okay. Let me make it a little cleaner gel. So this is a gel made up of agarose. Okay. Agarose gel. Like any other jelly toffees or gel that you have seen. It's exactly a gel. So agarose is it has long polymers like this. Okay. Once you heat it, it is liquid. You allow it to cool, it solidifies and makes a matrix, makes a gel-like matrix. Now, depending on what concentration of agarose, let's say I use 1%, which means 1 gram in 100 ml is 1% weight by volume. So if I make a 1% gel, it will have some molecules, right? Some polymers of agarose. Suppose I'm making 5% of agarose gel. Now, in this case, what happens? I will have 5 times more threads or polymers of agarose in it. Make sense? In the same volume? Now, pay attention here. When it was a 1% gel, it had some pores in between its polymers, correct? Can you see these pores that I am highlighting? When it is a 5% gel, more concentrated, what happens to the pores, average pore size? In which case the pores are bigger and in which case the pores are smaller?
In the first case, they're bigger.
When it was?
1 gram.
Less concentration means bigger pores. More concentration means smaller pores. Now the race is biased. Suppose I am making, and then of course you have to make wells, indentations in this gel, like potholes, and you load your DNA in it, your sample in it. Okay, you load these samples in the well. This whole thing is in a solution buffer, and you put this electric grid and you start the voltage. Okay, let's say I started at 40 millivolt, 40 volts or something. Yeah, 40 volts. Let's say the DNA will start running, but since they are running through a mesh of agarose fibers, and if it is a concentrated mesh, what will happen? Which kind of fragments will find it easier to move through the mesh and which kind of fragments will find it difficult to move through the mesh?
The bigger ones will find it difficult and the smaller ones will.
Exactly. So it is equivalent to like I'm making a deer with big antlers and a mouse move through a very bushy forest. Mouse will of course go through the bush within the bush and just clear the forest quickly. But the deer with huge antlers will get stuck, will try and move a little bit ahead and then again get stuck, will try and move a little bit ahead and again get stuck. So using the same voltage, the same electromotive force, but making them move through different mesh, different matrix, making them move through the same matrix. On the basis of their size, they will automatically move slow or fast. Yes or no?
Yes.
So that is the whole thing is gel electrophoresis. This is a gel. And you will get some bands. You will get one band here in your samples. Let's say you get bands here and you get a band here. Now what it means is this band is what? The biggest fragment or the smallest fragment?
Biggest.
Yes, this band is the largest fragment. And this band, which has moved the most distance in, let's say, half an hour, we run it for equal time, half an hour, we will get different fragments. This is the smallest fragment, right? Because it has moved the farthest in that mesh. So clearly it is smaller. Now, depending on how small your DNA fragment is, you can make a gel which is 1%, 0.5%, 2%, whatever you want, howsoever you want to make the pores bigger or smaller. But still, you will only know that this is smaller and this is bigger. You will not know which is yours. For that, in one of the wells, we always run a ladder. So these are preformed DNA sequences of different sizes that we run. This is like our control. And we know here that the highest one, it is a mixture between 100 base pair and the lowest one is 10 base pair. So the ladder is between 10 and 100, 10, 20, 30, 40, 50, 60, 70, 80, 100. Now, if you get your band at the same distance as of the 20 base pair ladder, you know that this fragment of DNA is 20 base pair. And if your gene of interest that you have calculated was also 20 base pair, this is your DNA fragment. Yes or no? There cannot be any other random 20 base pair fragment because you cut it specifically using a restriction endonuclease. And you know that it is going to be a 20 base pair or a 100 base pair or a 200 base pair or a 1000 base pair, whatever it is. Yeah. And using this method, you can isolate exactly your DNA. So what you have to do next is you will cut this gel using a blade, sterile blade. Cut this piece of DNA carrying your, cut this piece of gel carrying your DNA. Take this piece out like a burfi and then put it in a solution where the gel dissolves and all your DNA comes into the solution. That this is called DNA elution step. So you elute back the DNA from that gel and all that DNA fragments is your gene of interest. Then you purify that by a bait, putting a bait and they get stick to it. And then you ligate it with your plasmid vector. Make sense, everyone? Yes.
So the DNA are initially present in these cavities, right?
In these
So when you,
wells, yeah,
in the wells.
when we start,
So
yeah.
when you provide the voltage, is it like
Yeah.
the DNA goes up from these cavities and then rushes, the small fragments rushes at the end and the
Yes.
larger
So
fragment?
so this cavity is like the start line at the beginning of the forest, bushy forest. And everyone is standing there in the same well. A mouse, a deer with antlers, an elephant, a human, everyone. And then I say go. When I start voltage, you have to cross that, right? Because they are in a well, which means if this is the thickness of the gel, I am seeing the gel from a side now. And this is the well, okay? So your DNA molecules are all here, right? And the force is coming from this side. So obviously, to move, they cannot come out and start moving in the liquid. They will be pushed like this, right? They cannot be pushed up because the electromotive force is in this direction. If the force was from here, they could have pushed out of the well. But since it is not, they will be pushed like this. So the only option they have is to go through the mesh. That's why we make the well, to contain it within that gel. Okay?
Okay.
Make sense? Does it make sense, everyone?
Yes.
So it is nowadays the most commonly used technique where we use a matrix of agarose, which is a natural polymer, And agarose is extracted from seaweeds. These are plants present in the sea. And you can separate DNA fragments according to their size by a sieving effect provided by the electromotive force, electromotive gradient. So that's why phoresis, gel electrophoresis, you must have done something like this called chromatography. Have you done that? Chromatography? Paper chromatography, where you put a stain and different components of it separate as it move through capillary action. Yes, no? You have not done paper chromatography in your biology experiments? No. Okay, so just go back and look at chromatography and gel electrophoresis in on the internet and in the books. You will understand what I'm talking about. So that brings us to the last part of this chapter, which I will do in the next class. The next class, this chapter of principles will be over and we'll come to applications. In the next class, I will be talking about characteristics of a vector, where we'll talk about a vector called PBR3